Hello everyone. In this short video, let us learn the reason behind the uncouplers breaking the link between the electron transport chain and ATP synthesis. So before going into the actual reason, let us briefly discuss about the electron transport chain, how it is arranged and how it functions. So electron transport chain is a chain of protein complexes in the inner mitochondrial membrane. So they are arranged sequentially. So as you can see in this picture, so this is the inner mitochondrial membrane and this is the intermembrane space, that is the space between the inner mitochondrial membrane and the outer mitochondrial membrane. And this is inside of the mitochondria, that is called matrix. As you can see in this picture, there are mainly 5 protein complexes which are arranged sequentially in the inner mitochondrial membrane. So complex 1, complex 2, complex 3, complex 4 and complex 5. Apart from these complexes, 2 mobile carriers like coenzyme Q and cytochrome C also functions in the electron transport chain. So as the name suggests, what is the function of electron transport chain? It transports the electrons. So electrons are donated by the NADH and FADH2. These two are called reducing equivalents. So if you notice, the oxidation of different food molecules like carbohydrates, proteins and lipids, they produce NADH and FADH2, which are the reducing agents. And these reducing agents, they donate the electrons to the different complexes of electron transport chain, and then these electrons are carried at the complex 4, which are finally transferred to the oxygen to become water. So this process of transfer of electrons from one complex to another complex, ultimately to the complex 4, where it is donated to the oxygen to become water. This process is called as oxidation. This process is called oxidation. And if you see here, simultaneously when the electrons are being transported, in the first complex 4 protons are exported or pumped out of the matrix into the intermembrane space. Similarly, complex 3 pumps out 4 protons and complex 4 pumps out 2 protons. So when the transfer of 2 electrons are happening at the same time, 4 plus 4 plus 2, total 10 protons are pumped out if it is from the NADH. If the electrons are donated from FADH2, since it bypasses the complex 1, only the 4 protons from complex 3 and 2 protons from the complex 4 are pumped out. So total 6 protons are pumped out when 2 electrons are transported from FADH2. So when the electrons are moving from different complexes to complex 4 and ultimately to the oxygen, so simultaneously the protons are moving out of the mitochondrial matrix. Now as a result of this, there is formation of electrochemical gradient of protons. Now the proton concentration is very high in the intermembrane space and it is less in the mitochondrial matrix. And there is only one way by which these protons can enter the matrix is through the complex 5. So this complex 5 allows the protons to enter into the matrix. Simultaneously, there is generation of ATP from one molecule of ADP and inorganic phosphate. So the process which is happening at complex 5 is called as phosphorylation. So the process which is happening from complex 1 to complex 4 is called as oxidation, and the process which is happening at complex 5 is called as phosphorylation. So two processes are happening which are linked by the proton gradient, and hence oxidation and phosphorylation together called as oxidative phosphorylation. The same thing is shown in this picture, where the electrons from NADH and FADH2 are transferred ultimately to the complex 4 and from there to oxygen to form water. During this process, multiple protons are pumped out of the mitochondrial matrix, which leads to generation of electrochemical gradient of protons across the inner mitochondrial membrane. So the proton concentration is very high outside the inner mitochondrial membrane and it is less inside the matrix. So there is only one way through which the proton enters the matrix, that is by the complex 5. So when the protons enter through the complex 5, there is catalysis of generation of ATP. Combination of ADP plus inorganic phosphate leads to generation of ATP. So ATP is produced in the 5th complex. So this process is called phosphorylation and the transfer of electrons is called as oxidation. So oxidative phosphorylation is a coupled process. In fact, it is a very tightly coupled process. Now let us see what happens in case of uncouplers. So uncouplers are the substances that uncouple the oxidation and phosphorylation. So these substances allow electron transport chain to continue, but they prevent the phosphorylation of ADP and inorganic phosphate to form ATP. So oxidation will continue, but phosphorylation will not happen. So these are called as uncouplers. Since oxidation and phosphorylation are coupled process, these substances, they break the link between oxidation and phosphorylation, so they are called as uncouplers, and hence there is no ATP production, but the energy generated will be liberated as the heat in case of uncouplers. So these are some of the examples of uncouplers. The chemical uncouplers include 2,4-dinitrophenol, 2,4-dinitrocresol, chlorocarbonyl cyanide, phenyl hydrazone, and there are some physiological uncouplers which are already existing in the body, like thyroxine, bilirubin, and thermogenin in the brown adipose tissue. Thermogenin is a specific protein which is mostly found in the brown adipose tissue. And apart from this, some ionophores. Ionophores are lipid soluble compounds which can increase the permeability of lipid bilayers to certain ions. So valinomycin, gramicidin, these increase the permeability of the inner mitochondrial membrane for the protons. So these are examples of uncouplers. So now what is the exact mechanism of uncouplers? Uncouplers like 2,4-dinitrocresol, phenol, and even the thermogenin, they uncouple the oxidation with phosphorylation. That means oxidation from complex 1 to complex 4 is happening, which leads to generation of some free energy, and that free energy actually has to get converted to ATP through complex 5, because the complex 5 is the only channel for the protons to enter back into the mitochondrial matrix. But these uncouplers, they produce one more channel through which the proton enters the mitochondrial matrix. So this leads to the dissipation of the free energy as heat. So uncouplers break the link between the electron transport chain and ATP synthesis. The mechanism being, in oxidative phosphorylation, oxidation which is happening from complex 1 to complex 4 in the electron transport chain, and phosphorylation which is happening at the complex 5, they are tightly coupled processes. The link between oxidation and phosphorylation is the proton gradient. So this proton gradient is generated by oxidation, proton gradient is used by the phosphorylation. So the link between oxidation and phosphorylation is proton gradient. But what these uncouplers do? Uncouplers act through dissipating this proton gradient. So they provide another way for the protons to enter into the mitochondrial matrix. By this, even though oxidation is continuing, phosphorylation cannot happen. So thus it is known that uncouplers break the link between the ETC and the phosphorylation, that is ATP synthesis. Thank you.